to this. So go ahead and I'm going to actually ask Dr. Mead to do some introductory remarks, but I just want to let you know, just from a personal perspective, I am very familiar with the work done at the Nidegard laboratory. I have had the privilege of visiting twice Rochester for different reasons in the University of Rochester Medical Center, neurology and neurosurgery in 2 different occasions. And I have seen firsthand the extraordinary work that this laboratory does. And now when I was made aware of the work that Humberto, Dr. Mestre is doing, I was really, really excited about having, well, potentially figuring out how we can build bridges and understand more of this cerebral edema and how it relates to not just the things that he's studying, but also cancer, vascular issues, subarachnoid hemorrhage, a lot of things that pertain to this. So, Dr. Mir, would you like to say a few words, please, about Humberto?
Sure. Can you hear me?
Yes.
All right. Thank you, Dr. Q. It is a pleasure to present today's speaker, Dr. Humberto Mestre Payne. He's a medical doctor who graduated from Anahuac University in Mexico City. During medical school, he worked extensively at the neuroimmunology laboratory with Dr. Antonio Ibarra, developing models of stroke and spinal cord injury. He also did a couple of international rotations at the University of South Florida and Oxford University, studying these same topics. After graduating medical school with an impressive research background, he went to the University of Rochester in New York to the Division of Glial Disease and Therapeutics at the Center of Translational Neuromedicine for his Ph.D. As Dr. Q said, he worked under the guidance of Dr. Macon Nedergaard studying the lymphatic system. He is defending his thesis now in March, and he has applied to the match to start his neurology residency this cycle. Next, please. He has been working extensively in describing the lymphatic system with a special focus in vascular disease. Next, please. And as you can see here, he has made several contributions to science. He is co-author and first author of very impactful papers that have been published in journals such as Nature and Science, and sustains an H index of 20 in Google Scholar. On a personal level, I met Humberto when he was a when I was a first year medical student, and he was last year. He was actually my first mentor, and he was the one who introduced me to the world of neuroscience. We worked together in the neuroimmunology laboratory at our school for about 2 years, and working with him was just great. He taught me a lot about medicine and science with a lot of passion, but most importantly, he taught me about hard work, grit, and work ethic. He taught me to never give up and to never lose sight of what matters. As a mentee, it is very exciting to see all his accomplishments and to follow his path. And today he's going to share a little bit of the work that he has been doing during his PhD on the lymphatic system with a talk titled Cerebral Edema, as well as Time to Take Another Look. Welcome, Humberto.
And just to say a few words, because you won't hear from Humberto, from Paola, but I know that he is very actively getting a lot of phone calls from places like Harvard, Stanford, Hopkins for his residency in neurology. So Humberto, congratulations. I mean, to see the impressive body of work, the publications that you got in Mexico, even before coming to Macon's laboratory in very high impact journals, quite impressive. And many of us who understand science and the amount of effort that it takes, to produce these papers in parts of the world where the resources are not as rich as we have them here in the United States, is a testament of your extraordinary intellect. And I'm very excited to follow your career. And maybe one day we'll get you to be a faculty member here with us at the Mayo Clinic. Take it away, Humberto, please.
Yeah, thank you so much. That was an amazing introduction. And all I can really highlight is I completely echo what Paola was saying and the importance of mentorship. And I think I've been very lucky over the years. And so Paola, it's so nice to see you on this side of everything after all these years. And thank you so much for the invitation. So you're calling from the other Rochester. We're at 20 degrees Fahrenheit right now. So being in Jacksonville might have been really nice. But I always like to show this image here. This is coming at it from a completely different side of things. This is a Fiesta imaging showing all the free water compartments in the mouse brain. And so it very nicely kind of like pairs with the case presented this morning with the inner ear here, and we see the optic nerves connecting the eyes to the ventricular system in the brain. And so it just kind of highlights how underappreciated sometimes these large fluid compartments are to brain health within the neuroscience community. So I'm going to try to talk a little bit about what we've learned about all these fluid compartments and how they play a role in cerebral edema. So clinically, we were very interested in acute ischemic stroke. It's a very impactful disease with high incidence, high prevalence, caused by focal occlusions of cerebral artery, either thrombotic or embolic, and this causes a rapid reduction in the cerebral blood flow. And we know that one of the important complications of this is cerebral edema. And so in terms of cerebral edema, we get this abnormal accumulation of fluid, and we can see in this CT scan here, this hypodensity, and we get this mass effect with midline shift. And so we're trying to understand how we can potentially block our patients from reaching into these deleterious pathways, right? And historically, I think we've all believed that this is due mostly to vasogenic edema, or the abnormal entry of fluid from the vasculature. And so we call this a net fluid influx from the blood plasma. But we know that in the confines of the skull, this is quite a problem, right? Because we have a finite volume, and so we really can't increase a lot of that cerebral volume because it starts compressing on important structures like the brainstem that regulate our cardiac cycle and our respiratory cycle. So we really should develop new therapies to treat cerebral edema. We know that the clinical standard of treating a lot of this is either decompressive craniectomy, or we have hyperosmolar solutions like mannitol and hypertonic saline, but they really leave a lot of room to be improved in terms of, as long as we have this hyperosmotic gradient in the blood, we can actually remove the fluid and decrease ICP. But as soon as we lose that osmotic gradient, we have things like the edema returns, basically, right? And so we need to really understand what the biophysics of all of this are and the molecular mechanisms of edema formation to try to really develop novel techniques to treat it. And so historically, when we try to understand the phases of edema formation, especially after ischemic stroke, we have historically, Dr. Igor Klasko defined cytotoxic edema and then vasogenic edema. Cytotoxic edema being this very, very early onset of edema where we have cellular swelling, thus the name cytotoxic edema. But then several hours to days later, we have the onset of this vasogenic edema, which is where we have water coming across the blood-brain barrier and into the brain. And these were really considered separate entities for a long time. And I think clinically, sometimes we still see them as separate entities. But after acute ischemic stroke, some seminal work by Dr. Mark Simard and earlier found that maybe these two phases of edema are actually connected by an interim phase called ionic edema. And I think the most important thing to kind of get from this type of progression from cytotoxic to vasogenic edema is that the edema content actually follows the cation content. Primarily, the cations that we're interested in are sodium. Because sodium will generally pull chloride, and when sodium and chloride move across membranes, they basically pull water. And so you'll notice that edema really tightly follows cation content. And so this will be important later on in the talk. And we know that if we have capillaries seen here as cross-section, and then we have sodium ions kind of floating inside the blood vessels, and then we have neurons sitting outside these capillaries, we know that during cytotoxic edema, when we lose the ability to produce ATP, cells are no longer able to kind of power their sodium-potassium ATPase pumps. And so they basically depolarize, letting a lot of sodium into the cell. And this depletion of sodium within the extracellular space actually causes sodium to enter across the capillary wall. And so we think that this is driven by the upregulation of de novo channels like SIR1-TRPM4. This is Dr. Simard's work. And this is the case of why we can use drugs like glyburide and like the CHARM trials to prevent edema formation, because they basically block these SIR1-TRPM4 channels that allow the entry of sodium from the blood into the brain. So tackling this cation content accumulation. But we know that once we have this cytotoxic phase, we also have these later ionic edema phases, which is where we get this abnormal entry of ions into the brain tissue. But then later stages, very important, vasogenic edema and the entry of water across disrupted blood-brain barrier or transport across the endothelial cells. And then eventually we get to the point where the inflammatory response and the expression of matrix metalloproteinases can basically destroy the capillary wall, and then we have the entry of blood into the CNS. And so this is probably the much later stages and complicated evolution of acute ischemic stroke, but still important nonetheless in terms of understanding edema formation. So we know that edema correlates with sodium content, and we know historically that blood-brain barrier actually occurs up to several hours to days after a stroke. And so it doesn't really happen in the very early time points after stroke, but we already know edema starts very early. And one thing that we hadn't studied was that CSF is actually quite high in sodium concentration, similar to plasma. So we just asked the question of whether or not CSF contributes to edema formation, mostly because we work with this glymphatic system. And the idea that CSF is propelled into these periarterial, perivascular spaces into the brain via these Virchow-Robin channels and across the astrocyte end feet via these Aquaporin IV water channels. And this CSF exchanges with the interstitial fluid, and then it's pumped downstream. And this process plays a role in removing metabolic waste from the parenchyma, like amyloid beta and tau. And then that fluid is pumped down to perivenous spaces, and then is pushed through dural lymphatics out into the periphery. And so we call this is Macon called this the glymphatic system because it's glial lymphatic system of the brain, or glymphatic system. And so we are interested in this, in understanding how the system works. And if we know that the ischemic brain is basically thirsty for taking on abnormal fluid amounts, then why isn't the CSF that's getting pushed in via these periarterial spaces driving some of that edema fluid? So one way that we have to evaluate these glymphatic flows is by using animal models. In this case, we can anesthetize a rodent, and we can put a small cranial window over the skull. And then we can actually inject fluorescent tracers and use 2-photon imaging. And what you get is you label the blood vessels as well. And so you see these CSF tracers kind of entering along these surface periarterial spaces, and then they dive down to subcortical structures via this penetrating arterioles here. And so we can use this technique, but unfortunately it doesn't really give us a ton of information in terms of the flow, because we can only see this fluorescent tracer front kind of moving through the brain surface. And so one thing we can do is we can convert this into particles like this, and we can record the ECG of the animals, or the respiration of the animals. And using a particle tracking velocimetry software, we can actually measure the pulsatile frequencies of CSF, seen here in these little green dots, together with the heartbeat of the mouse. And so what we found is that every time we get this beating of the heart, we get this pulsatile flow of the CSF in these surface periarterial spaces deep down into the brain. And so we know that these arterial pulsations are very important in driving CSF flow. So our first hypothesis was that if you cause acute ischemic stroke and you completely destroy these pulsations, then we should get no CSF being propelled into the brain tissue. And that was kind of our working model initially. And so that's how we decided to approach this. And so, you know, this is how we can evaluate how important the arterial pulsations are. Over a 30-minute experiment, we can basically overlap every single particle track of a 30-minute experiment. We get like 20,000 particles, and we overlay them over these periarterial spaces. And then we can calculate the flow speeds within these surface spaces. And you'll see that it seems very heterogeneous, with like parts where it speeds up and then parts where it slows down right here in the middle.
And Humberto, just a quick question about this, the actual experiment itself. You say you marked this with nanoparticles, or how was that? How do you mark the CSF flow? I was looking at those green dots. I wasn't quite sure I understood that part of the methodology.
So these are 1 micrometers fluospheres that you basically inject into the cisterna magna.
Gotcha.
Yeah.
Gotcha. And then you can track them so they are fluorescent, and then you can, and you're doing an open window on the skull of the rodent, I take it, and put in
So,
the the multiphoton.
yeah, so but we do we basically seal the craniotomy again with a cover slip to kind of prevent the intracranial depressurization, because we know that that affects a lot of this pulsatility. So
Gotcha.
they're resealed cranial windows.
Beautiful.
Yeah. And so basically, if we record the ECG of the animal seen here and the respiration of the animal, and basically look to see what the CSF is doing after every heartbeat or after every breath, and we see that after every heartbeat, we get this nice reproducible increase in pulsatile flow, about 35 milliseconds after each heartbeat, which suggests it's the time it takes for the pressure wave to reach the heart to where we're imaging in the brain. But when we look at respiration, there's really no modulation. So this is very strong evidence that it's this arterial pulsation that's driving the flow. Although measuring the ECG is an indirect metric of this. And so one thing we can do with the 2-photon is we can take these line scans. And basically what this gives us is the ability to record the ECG and measure the diameter of the artery as we're imaging. And so once we do this, and we plot basically the derivative of this artery diameter just to check how quickly that wall movement is happening. We can plot that seen here as wall velocity. And then we can overlay that with this VRMS, which is the CSF tracer. And so we see that the peak of when the particles are pulsating and the peak of when the wall is moving is actually at the same time. So suggesting that these arterial pulsations are actually driving the CSF flow in these periarterial spaces. So this takes us back to our clinical interest, which is acute ischemic stroke, and wanting to evaluate what happens with CSF flow after a stroke. But we really wanted to look at CSF flow right at the moment when we were having these strokes. And so we didn't want to use a filament model where you're basically having to put the filament in while the mouse is under the microscope. You have to wait a couple hours and then pull the filament out, suture the animal, put it back under the microscope or in the MRI magnet. And so what we wanted was an embolic model where we basically were able to image before, during, and after the stroke. And so the way we did this was by infusing a 200 micrometer macrosphere into the common carotid artery. And luckily, these spheres also, similar to the human anatomy, they become lodged at the base of the MCA. And so it's very similar to an M1 large vessel occlusion. And then you can see the sphere here at the base of the MCA. We can use TTC staining to show where the infarct is 24 hours later. And so white is infarcted tissue and pink is live tissue. And so we get a pretty reproducible infarct that spans most of that ipsilateral hemisphere. But the most important part is that we have tight temporal control over when we induce a stroke. And so if we measure cerebral blood flow using laser Doppler, you can see that when we infuse a microsphere, we get this rapid 92% drop in cerebral blood flow. So we have good temporal control over when the stroke happens. And so this gives us the ability to basically image the animals and label their CSF as we're inducing a large vessel occlusion. And one way we can image CSF is by injecting fluorescent tracers into the cisterna magna. And mice have a very, very thin skull. And so we can actually image through the intact skull by just reflecting the skin. And we can use tracers that are far red fluorescent. So they have great, they have very low tissue absorbance. And so we can get very good imaging. So I've overlaid the bone structure here on the mouse, and this is what it's going to look like. And we inject a CSF tracer, and you can see it distributing, and then we induce a stroke. And a couple minutes after, we get this rapid influx of these CSF tracers across these surface periarterial spaces of the ipsilateral hemisphere. And we don't get the similar pattern on the contralateral hemisphere. So there's a lot of CSF entering on the side of the stroke. And we can plot this data together with the CBF just to show that in every single case, we get this rapid drop of blood flow. So these are all good strokes, and the blood flow doesn't return during the time of the experiment. But then we can quantify the fluorescence of the ipsilateral hemisphere seen here in red and the contralateral hemisphere seen here in black. And so we see that in every experiment we did, we get this rapid influx of CSF to the side of the stroke. And we can get the derivative of those curves to show when most of this activity is happening. And we see that we get the small peak right after we induce a stroke, but about 5 minutes later, we get this rapid influx of CSF. And that's going to be important further down the line. But what's really important to us is edema. And so one way that we can quantify edema is by basically taking the brain out after 15 minutes, weighing the wet weight, putting it in an oven for 48 hours, desiccating the tissue, expecting all that water to evaporate off, and then weigh the dry weight and basically subtract those two to get edema content. And so we noticed that by 15 minutes, we already have edema formation within that ipsilateral cortex, which is when all the CSF is entering the brain tissue. So there's already edema and there's a lot of CSF going in. So it just begged the question of whether or not CSF is contributing to this edema formation. And as I mentioned, there's no really great way to evaluate water by itself in terms of tracing it. But what I mentioned was that cations seem to be very important as to where the water is going and where it's coming from. And so one way we can do that is by labeling the cations. And so we can use radiolabeled isotopes like 22 sodium. And we can basically inject the 22 sodium either into the CSF and then quantify it in brain, or we can label it, or we can put it into the bloodstream and then label it in brain as well to measure the influx of both compartments into the brain tissue. But then we can also use other radiolabeled markers like tritiated mannitol. And tritiated mannitol has the interesting advantage that it shouldn't really cross cell membranes. And so, for example, if we see the entry of mannitol from blood into brain, then maybe this is a marker of a permeable blood-brain barrier, right? And so if we don't see any entry of mannitol, it might suggest that our BBB is still intact at these early time points. And so for the first experiment, we inject these radiolabeled tracers into the blood, induce a stroke, and then quantify 15 minutes later. And what we find is that we don't really see a difference between the amount of sodium entering the ischemic hemisphere seen here in red, and then the normoxic hemisphere seen here in left. So there's really no difference in terms of sodium and mannitol. However, when we do the converse experiment and inject both the tracers into the CSF pool, we actually do see a significantly larger amount of the sodium in the ischemic hemisphere and even the mannitol in the ischemic hemisphere. So suggesting that most of the sodium and perhaps most of the water that's causing the edema within the first 15 minutes is coming from the CSF.
And Humberto, just a quick question. I mean, you may tell us something different, and I'm excited about what I'm about to hear from you, hopefully. Wouldn't the CSF flow into these areas be nothing else but a mechanical property of the path of least resistance? When you introduce the occlusion of the MCA, vessels decrease in size, and now you have the space between the glia and the blood vessels increase, and then CSF sort of flows. I mean, that's a simplistic way of looking at this. Maybe CSF is smarter than that, but
that's exactly what we thought. That's what we thought. We thought that it was basically a very simple kind of loss of pressure phenomenon.
Yeah.
But if you'll notice from this trace here, it actually takes about 5 minutes for the CSF to really enter the brain. So if you'd expect it to be just like a hydrostatic change, you'd expect it to take place within the first couple seconds. And so it's a little bit more delayed than we expected. And
Gotcha.
I think the story gets a lot more interesting. And so
Gotcha.
I'll show you some of that.
All right.
But so, we love to image rodents in all shapes and ways. One of them is using rodent MRI scanners. And so what we did here was we wanted to use the same kind of tracer techniques using dynamic contrast enhanced MRI. We can inject gadobutrol into the CM just like we do fluorescent tracers. But then we can also do diffusion weighted MRI to test or evaluate this edema formation to see whether or not these processes are happening at around the same time. And so you'll see we start inject the fluorescent, the gadolinium-based tracers into the CM. They start spreading around, and then we induce a stroke here, and you'll see a spreading wave of cytotoxic edema. This is diffusion restriction here. It kind of spreads throughout the ipsilateral hemisphere. But one thing you'll notice is that the CSF tracer is accumulating throughout this ipsilateral hemisphere, and I can kind of show the spreading wave again. And so you get this wave of depolarizing cells. It's the onset of cytotoxic edema, and then you get the CSF entry. So this goes back to your point, Dr. Quiñones. This spreading wave of cytotoxic edema was really what triggered the influx of CSF. And so one of the issues with the MRI technique is that we can't really image both sequences at the same time. And so we really wanted to evaluate both the spreading wave, and I'll get into what that is, but also CSF tracers. And so this spreading effect, we didn't know at the time, but they're known as spreading depolarizations. And these were described by Liao in the 1950s, this phenomenon in which cells basically lose the ability to maintain their ion gradients and in one cell depolarizes, basically causes a subsequent cell to depolarize in the subsequent, and you basically get this domino effect throughout the entire brain tissue. And so you get this wave of depolarization. And this is believed to be the onset of cytotoxic edema, that first harbinger of edema formation. And so we can use genetically encoded calcium indicators like GCAM seen here, and we can also label the blood vessels. And we can use 2-photon or macroscopic imaging techniques to see these waves, these spreading depolarizations. And we worked with Dr. Hash Hirase, who was at Riken at the time, and he developed these amazing GLT1, which is glutamate transporter 1, GCAM7 mice. They express this GCAM almost throughout all of cortex. It's expressed in 100% of cortical astrocytes and about 60% cortical neurons, seen here with Nguyen staining. And what's great about this is that the fluorescence is so bright here at the surface that we can actually use that technique that I was showing you before and image through the intact skull, but also pair it with CSF tracers at the same time. And so the imaging looks a little bit like this. So we do the same CSF tracer injection, we induce the stroke, and about 5 to 7 minutes later, we get the spreading depolarization. And right after that spreading depolarization, we get this rapid CSF influx. So once we quantify this data here, we can see that the spreading depolarizations actually happen in every single mouse we did in the ipsilateral hemisphere. They happen at variable times after the stroke, but the onset of the spreading depolarization actually varies from mouse to mouse, and the literature says that it has to do with how high the blood glucose is, or how long their last meal was before the day of the stroke. And so this is what really determined the onset of when the CSF influx happened, as opposed to that rapid decrease in blood flow to that ipsilateral hemisphere. So we can quantify the CSF influx just as we did before, but the timing kind of like shifts when the CSF influx occurs. So if we align every single CSF trace to when the spreading depolarization occurs, every single time we had a spreading depolarization, we had CSF influx. And so it's this wave that really triggers the entry of CSF and not just the loss of blood flow to that area. But one thing we did notice was that even though we had the spreading depolarization, it still took about 10 to 30 seconds after the spreading depolarization for the influx to happen. And so we wanted to evaluate this closer. We wanted to use 2-photon. And so similar as before, we can do this type of imaging where we can label the PL arterioles, and we can look at these penetrating arterioles that go down into the brain. This is basically a volume. Here you're looking at from the top, and here you're looking at from the side. And so this is the brain surface and the blood vessels diving down here. So we can image this by injecting the fluorescent tracers into the blood to look at the blood vessels, the CSF tracer seen here in green, but we can also image these GCAM mice. And so we have 3-channel imaging, and what you'll see here is the spreading depolarization washed through the cortex, and then you see entry of CSF tracer down through these penetrating arterioles that are diving deep into the brain. And this is a better image of what that looks like, but you see the spreading depolarization, vasoconstriction, and then the entry of CSF into the brain. And what we know is that these vasoconstrictions are actually called spreading ischemia. And similar to neurons in the brain, smooth muscle cells are also basically polarized, or they can depolarize essentially. And so basically smooth muscle cells are no different to neurons in that when all the cells are depolarizing with the spreading depolarization, they also depolarize. And so you get this rapid vasoconstriction of all the arteries in the brain. And that's what we think is driving the CSF. And so we get these spreading depolarizations here, then you get this arterial diameter decrease, and then you get the CSF influx. So we really think it's the changes in vessel diameter and not the spreading depolarization by itself that's driving the CSF. But we really, we have an issue because we're outside the resolution of MRI, for example, to look at these small surface arteries. And 2-photon is great, but it gives us like a 1 millimeter by 1 millimeter window. And so we can't really evaluate what's happening with the entire vascular network throughout the brain hemisphere that might be driving the CSF into the brain tissue. And so we worked with David Kleinfeld and Pablo Blinder at UCSD in Tel Aviv, and we worked with some mathematicians at Danish Technical University, and we basically used these geometries of the entire Peel middle cerebral artery of mice. And so basically they just take a brain, squish it down, and we can generate this 2D network of what the entire MCA distribution looks like. And what we can do is we can basically model what the spreading depolarization looks like as it's crossing the entire parenchyma. Then we get these vasoconstrictions that increase the size of the perivascular spaces and how much fluid do you need to insert into this network to kind of facilitate that spreading wave of CSF influx. And so this is the area of the spreading depolarization over time. And this is the area of these perivascular spaces over time. And so we can expect to see about a 2-fold increase in the amount of CSF flow at this inlet of the MCA to drive all the edema that we're seeing in these rodents. And so the only way to have really useful models is to be able to experimentally test whether or not that's the case. And so we can use that particle tracking velocimetry technique that I mentioned before. And in this case, what we've done is that we've labeled the particles by how fast they're moving. And so one thing you'll notice is at the beginning, it gets very sludgy. We have very little pulsations. There's very little CSF flow after the stroke. But then at about a couple minutes after, you start getting this rapid inflow of the particles through these periarterial spaces. And you also see that the artery kind of decreases its diameter, so the spaces become larger, and you get this large inflow of CSF. So when we quantify the flow change, we see here the change in artery diameter after the spreading depolarization. But if we quantify this V downstream, which V downstream is basically how quickly these particles are traveling into the brain here, we see about a 2-fold increase in the flow speed. So it really does validate our modeling results. So we should expect about a 2-fold increase in flow speed in a realistic MCA model. So we do think it's this spreading ischemia that's kind of driving the CSF inflow. And then we're able to kind of quantify edema formation in different types of tissue. And so you'll see here that in mice, we can do just H&E stains to look at how edema is forming in different areas. And so we see that at the surface of the brain near the leptomeningeal vessels in the stroke animals, we get this with this impressive amount of edema formation. And this is areas that are near CSF, right? And so if we go to striatum, we don't see the same amount of edema formation in the early time points. But if we go to ependymal areas next to the ventricle, which is another pool of CSF, we do see edema formation. And we saw a same feature in human brain samples that had undergone ischemic stroke. And so suggesting that areas that are nearby CSF pools swell earlier than areas that are farther away from CSF pools. And clinically, what we're interested in is developing new techniques or new therapeutic interventions. And so one way that we can do this is by blocking Aquaporin IV and knocking Aquaporin IV completely out in these rodents. And when we do that, we see tracer entering in the wild types diving down into the brain. But when we do that in the knockouts, we see very slow CSF entry and very little actually dives into the brain. And so We can stain the tissue here for Aquaporin IV and look at the CSF tracer seen here in magenta. And we get a lot of CSF tracer entering the brain in the wild types, but when we knock Aquaporin IV, we see almost no tracer entering the brain. And this actually correlates with a decrease in edema formation in the knockouts. And so very interesting, the idea that we could potentially use Aquaporin IV inhibitors to kind of prevent this very early edema formation after stroke. And so basically, this is what we're thinking right now, is that glymphatic influx is one of the earliest contributors to edema formation. We think that these spreading depolarizations are what's triggering the CSF influx. And what we're finding is that CSF influx actually delivers cations. And these cations basically correlate with how much ionic edema and how much vasogenic edema we might get later on. So we're interested in evaluating how this very early process might worsen the later stages of edema formation. And then, of course, the exciting avenue that blocking Aquaporin IV might be a very promising treatment for preventing this early edema in these patients. And so this is a gargantuan effort by a lot of people. This is all spearheaded by Dr. Niedergaard's work, both labs in Rochester, New York, and Copenhagen. Our collaborators that work with fluid dynamics who help us with a lot of the particle tracking velocimetry. And we have mathematicians and blood flow specialists at UCSD, and of course, acknowledge a lot of our funding sources.
Beautiful work, Humberto, beautiful work. If you can go back to that slide for the mechanisms, the methodology, I'm sure we have some questions already coming up. And just to kick it off with one potential thought, we're beginning to understand, thanks to the work in your laboratory with Macon and some of the work out of UCSF as well, a number of years ago with Aquaporin IV. This when I was a resident, actually. The first author of that paper was Jeff Manley, and then Devin Binder, that were working with the group there. So there's been a tremendous advance, but when it comes down to, imaging the brain with the multiphoton, your team has been really at the forefront in the world. And we're beginning to understand stroke, as you said, and I think that is very sort of encouraging to see this. I wonder then, how will this translate to other pathophysiological events like brain trauma, of course, being an important one. And the other one for us, for me particularly, is brain cancer, which also produces a fair amount of edema. And then I'll kind of open it up for the audience.
Yeah, I mean, it's very exciting. I think that, for example, in the case of tumors, right, we know that there's a lot of the cytotoxic edema, and I think that there's a lot of work into trying to understand how tumors might disrupt the blood-brain barrier, and so is the fluid coming from the vasculature. But we also know that a lot of these tumors are in close vicinity to CSF pools, and so if we do kind of have this abnormal glymphatic fluid entry that basically drives edema whenever you kind of have either decreased ATP production from these tumors or just a lot of pro-inflammatory markers that might open the blood-brain barrier, there's options to kind of like try to tackle some of these glymphatic related causes of edema. And so I think that there's a lot of exciting opportunities in that sense. I mean, there's also, not to mention the fact that these spreading depolarizations are not only exclusively found in ischemic stroke, but we have them in subarachnoid hemorrhage and TBI. And so we have a lot of these patients in our neuro ICUs, and our continuous EEG is basically filtering out a lot of these spreading depolarizations. And so some of the work coming out of Jed Harding's lab, Jens Dreyer, and the Cosbit group trying to like measure the amount of these spreading depolarizations in the neuro ICU, because they might be contributing to edema formation, and we're not really like tackling them. And so I think it kind of opens interesting opportunities to kind of measure some of this phenomenon.
Dr. Barrett, from the stroke perspective, I saw your comments, and then I'm going to see if Dr. Tatum is there from the epilepsy perspective because of all these depolarizations and edema. Go ahead, Kevin.
Yeah, this is excellent work. Congratulations on putting this together so clearly for us. The animal model, and some of what we focus on in the acute ischemic stroke practice is with large vessel occlusion, but even at the level of second, third order, or end artery ischemia, we see patients who have neurologic deterioration that may occur a day or two after a lacunar infarct. And so, you know, this is occurring at likely all levels. And I think the large vessel occlusion model is probably just the beginning of our ability to not only understand, but explore the translational aspect with the Aquaporin IV inhibitors and other approaches. So this is great.
Yeah, completely agree. We chose a 200 micrometer sphere, but we can probably go a lot lower and evaluate, you know, models like lacunar infarct as well, which are really
And how feasible it is, Dr. Barrett. So Humberto, just so you know, Kevin is the chair of neurology here at Mayo Clinic in Florida. So how feasible it is? Are there medications already, Kevin, for Aquaporin IV that are FDA approved or used for some other diseases that we could potentially envision in 5-10 years down the line being able to use here?
Yeah, certainly the importance of the Aquaporin IV has become evident in autoimmune and demyelinating conditions that affect the brain. So this is an area definitely with overlap and potential for therapeutics.
That's great.
There's a phase 1 clinical trial right now by a company, I think it's a spin out of Yale, Aeromics, who's testing a brand new Aqp4 inhibitor. So 5 years down the line might be realistic. That's exciting.
Amazing. And then I don't know if Dr. Tatum, are you still there on the line before epilepsy? Maybe he left us already.
I have a question, Dr. Q.
Okay, Ana, Dr. Carrano, please.
Humberto, so you measure the contribution of CSF flow and the blood at, I believe, 15 minutes. Are these effects sustained over time, or I mean, did you perform this experiment for longer times?
Yeah, that's an excellent point. No, so for this model, we were kind of worried about the fact that these were embolic strokes. And so we were basically infusing this large sphere, hoping that it becomes lodged, but then have no control over whether or not it becomes dislodged. And so we were a little bit hesitant to kind of study the animals out to 24 or 48 hours, not knowing whether or not it'll become dislodged or not. And so for now, we were only looking at the very acute time points, but that's definitely something that we're interested in looking down the line.
Okay, thank you.
Thank you, Ana. Any other questions? I see a comment right here by Dr. Vargas from Colombia, you know, and congratulating you, spectacular work. You know, brain tumor edema is one of the main problems of our patients, and how to stop that maybe help a lot. But, and I was thinking even beyond that, I was looking at reminiscent about some of the work of Skip Grossman out of Johns Hopkins, where the very first papers that they talk about the lymphatic system in the brain, he began to hypothesize that maybe this cerebrospinal fluid flow was also the corridors that cancer cells may be using to migrate and to move from one place to another. And when I see your pictures and your movies, it makes perfect sense that there's flow, there's movement, there are corridors that we still don't understand. And they're probably different. Every brain is going to have different roads. And I think that your MRI images and your images of flow is beginning to give us some insight potential as to how this edema may be contributing to the spread of the cancer. And we don't even understand it yet.
Actually, I have a
Emily.
comment. Yeah.
Go ahead,
kind
Emily.
of related to that. There was a recent paper, I don't know if it was last month or earlier this month, but describing that increased CSF and peritumoral edema is associated with worse prognosis for the patients, which kind of validates the role of the lymphatic system and you know, swelling, as well as the role of just, you know, we studied the CSF, so the role of the CSF in the tumor biology. So I think it would be really interesting going forward to kind of, you know, tease out those mechanisms in brain tumor biology.
Well, this is what we wanted to build bridges with Humberto and make. And I went and visited her lab back in 2007, and then went back in 2010 with her and Steve, who's in the committee for Humberto's Ph.D. I think I'd love for us to maybe invite you, Humberto, to help us kick off our multiphoton microscope understanding of a lot of these issues, in a collaborative effort, because we're really excited, not only from a stroke, as you know, I mean, with Dr. Barrett and Dr. Mestre and Dr. Brott, they have an amazing clinical NIH grant for stroke, and we're very much, and I saw Michelle Lin right here also, she's got an NIH grant also. So there's a lot of interest in stroke, but also from the brain cancer perspective. Emily mentioned that she studies the CSF role, the biology, the interaction, but we still don't understand the mechanics. But those mechanics are not driven in isolation in a simplistic way. It's got to be some pathophysiology. And then you integrate that with seizures and all these depolarizations. Now you have a very complex system, and how to break it down for cancer is going to be probably the next frontier for us, undoubtedly. So you have an open invitation. We'll work. Dr. Mead, any last words? I know that we went over time, but any last words for your mentor?
Yeah. First of all, congratulations. It was an amazing talk, Humberto. And Dr. Yu, I think there's also another opportunity to study the lymphatic system in tumors with the work that we're doing with Dr. Schiaparelli in regards to NKCC1 and the small molecule inhibitors that we're testing. So that's one of our special interests. We can talk with Humberto a little bit more offline, but it is, we think that cell swelling and migration is definitely one thing to look into.
Beautiful work. Great. Well, Humberto, thank you very much. We will send you a something from our department. This was an amazing, impressive body of work that you personally have done, but also in Makin's lab, and it's very, very nice to see it. We're very honored to have you here today, and we're looking forward to seeing you in person. Hopefully, the restrictions for traveling will diminish soon.
Thank you so much for having me.
Congratulations. Have a great week, everybody. Bunch of conferences. We'll see you guys on Friday. Take care. Bye-bye.
Bye.